The role of the liver. The role of liver. Many food molecules absorbed into the blood intestine are carried to the liver for assimilation, conversion of food molecules to other molecules that the body needs, such as fibrinogen, a protein found in the blood plasma that is important for blood clotting. Excess amino acids are absorbed into the blood that is not needed to make proteins. But they can't be stored. They are broken down in a process called deamination. Deamination is the removal of the nitrogen-containing part of amino acids to form urea. Enzymes in the liver split up the amino acid molecules. The part of the molecule which contains carbon is turned into glycogen and stored. The other part containing nitrogen is turned to urea. The urea dissolves in the blood and is taken to the kidney. A small amount is excreted in sweat. The kidney, located in the back of the abdomen. They regulate the water content of the blood. They excrete the toxic waste of products of metabolism and substances present in excess. Each kidney contain 1,000,000 tiny structures called nephrons. The nephrons in the cortex of the kidney loop down into the medulla and back up to the cortex. The contents of the nephrons drain into the innermost part of the kidney, and the urine collects there before it flows into the ureter, and it flows into the ureter to be carried to the bladder for storage of urine. Processes in the nephron. Ultrafiltration. Arterioles branch off the renal artery and lead to each nephron, where they form a knot of capillaries sitting inside the cup-shaped Bowman's capsule. The capillaries get narrower as they get further into the glomerulus, which increases the pressure, already at high pressure. This eventually causes smaller molecules being carried in the blood to be forced out of the capillaries and into the Bowman's capsule, where they form what is known as the filtrate. The substances forced out are glucose, water, urea, salts. Some of these are useful and will be reabsorbed back into the blood further down the nephron. Reabsorption of glucose. After the glomerular filtrate enters the Bowman's capsule, glucose is the first to be reabsorbed at the proximal convoluted tubule sagittar. This takes place by active transport. Nephrons have many mitochondria for this process. Reabsorption of glucose cannot take place anywhere else in the nephron. Reabsorption of water and salts. As the filtrate drips through the loop of Henle, necessary salts are reabsorbed back into the blood by diffusion. As salts are reabsorbed back into the blood, water follows by osmosis. Water is also reabsorbed from the collecting duct in different amounts depending